Welcome to the neuronal action potential video. And so, first of all, if you want access to this worksheet, there's a link with the download PDF available on Canvas. And so, first of all, make sure you have this printed out. And second of all, maybe get a lot of different colors of pens to start off with. I'm personally going to be using all these different kind of colors. So I got pink, red, black, purple, 2 different colors of blue, and I'm also using a pencil. Hopefully the different colors can make things a little bit more clear. And so once you have those materials, we can get started on firstly discussing how is the inside of the neuron different than the outside of the neuron. And for reference, where is the neuron in this picture? It is the structure ranging from this area, and including this area, all the way down here to the end. And they have different parts. We have the dendrites in the beginning, we have the axon hillock, then we have the axon, and we have the axon terminal. And this axon terminal is adjacent to another set of dendrites from an adjacent neuron. Similarly, we have adjacent neurons' axon terminals right here, next to this dendrite. So, first things first, let's talk about specifically neuron cell gradients. So, whether it's a neuron or not a neuron, all of the cells in your body are going to have a similar kind of style of gradient, which is going to be sodium, higher concentration on the outside, and potassium, higher concentration on the inside. I like to think about our cells in the body kind of like a banana in the ocean. So we think of bananas as having high potassium on the inside, right? That's very similar to our cells. Our cells have high potassium on the inside. If we think about that banana being submerged in the ocean is very salty, high sodium on the outside of the banana. So what establishes this gradient? Well, I'm going to draw your attention to this area of the worksheet. So there's a couple different membrane transport proteins drawn in here. And by the way, this is the cell membrane of the neuron. So you'd find these particular membrane transport proteins anywhere along the dendrite or the axon hillock or the axon or axon terminal. Maybe not in this part of the axon. We'll talk about that later. But anyway, we got this membrane transport protein here. This is called an NKX, or a sodium-potassium pump. Its job is to pump 3 sodium ions into the extracellular matrix. We can call it the extracellular fluid, or just the outside of the cell. We can kind of simply draw that by drawing an arrow to the outside of the cell, and just 3 Na+. And I'll zoom in here for you guys. There we go. So when this sodium-potassium pump works, it is going to transport 3 sodium ions out for every 2 potassium ions in. So 2 potassium ions into the cell. So you can imagine if this keeps going and going and going, we're going to end up with a ton of sodium ions on the outside and a ton of potassium ions on the inside. The really important thing to take away from this is that gradient and also the electrical gradient. Because if you think about what is the consequence of pumping 3 positive ions, whatever they are, out versus 2 positive ions in, whatever they are. If we have 3 positives out, 2 positives in, that is going to create a difference of 1 positive charge across the membrane. If you pump this pump twice, that means you now have 6 sodiums out, 6 positive ions, and 4 potassiums or 4 positive ions in. That's a gradient of 2 positive ions. So as this builds up, we end up with a difference of really positive charge on the outside of the cell and a relatively negative charge on the inside of the cell. And that is a big concept in action potentials, is that we start off, our resting state starts at the cell being negatively charged on the inside and the cell being positively charged on the outside. So one way we can write that is, just remind ourselves with maybe some pluses on the outside, just outside of the phospholipid bilayer, and we can add some negative charges on the inside of the cell. So this would be where the cytoplasm would be of the neuron. Now there's another additional thing that is going to make the inside of the cell negatively charged. We not only have that difference, but we also have proteins inside of the cell that have negative charges, and those large proteins contribute to the overall negative charge of the inside of the cell. We also have your DNA or RNA, which have negative charges. And so let's just summarize DNA and RNA as nucleic acids. They also will have a negative charge, so they contribute to that as well. Now this part, we're not going to get into the rest of these channels. I don't want to get into too much detail about them, but if you ever take more advanced neurophysiology courses or neuroscience courses, it might be useful to know that there are these other leaky channels in the membrane known as potassium leaky channels, which are both of these potassium leaky channels, where the potassium can diffuse back out of the cell. And we also have sodium leaky channels, which we have fewer of, hence I drew just one of these versus 2 potassium channels. So we have fewer sodium leaky channels. That allows sodium to go from the top back into the cell. So even though those leaky channels exist, the gradient is still going to be mostly sodium on the outside and mostly potassium on the inside. Okay, so now that we have that established, we can start to talk about the steps of an action potential. Oops, just let me move this. Okay. So step 1, let's start actually with this axon terminal from another neuron. In this axon terminal, and let me zoom in as well. Okay. So in this axon terminal, it has these structures called vesicles filled with neurotransmitters. So these are called synaptic vesicles. And these synaptic vesicles, I'll spell it synaptic vesicles. And they have neurotransmitters. I'm just going to say NT, and NTS for plural. And this vesicle can fuse to the cell membrane of this neuron's axon terminal, and when it fuses, it can release those neurotransmitter contents into the synaptic cleft, the space between this neuron and this neuron's dendrite. These dark triangles represents a ligand-gated sodium channel. Ligand-gated sodium channel. Let's go ahead and make a key for this stuff. So I think it might be a good spot to make a key here. Where we say there's a dark triangle, and that dark triangle represents a ligand-gated sodium channel. And so ligand-gated sodium channels, essentially what that means is that the sodium channel is only open when the proper ligand attaches to it. And the word ligand is pretty much a term that describes any sort of molecule that it's receptive to. So in particular, let's talk about an example for a common example. A common ligand would be acetylcholine, or ACH. And so ACH, or acetylcholine, would be the neurotransmitter, and it is also considered a ligand. So that neurotransmitter slash ligand is going to bind this ligand-gated sodium channel. So if you imagine those neurotransmitters get released into this space, they activate these, and when they're activated, they actually physically will open up so that there's a pore in the dendrite of the neuron. So that pore opens up. That allows sodium to go into the dendrite, into the cell. And what is sodium doing on the outside? Why isn't it just, where did this come from? Remember, sodium comes from this pump. It gets in high concentrations on the outside of the cell. So we have a lot of this on the outside, only a little bit on the inside, and it has a gradient. It would like to move down and enter the cell. So this is a passive sort of diffusion. Now, maybe you can imagine there's all this neurotransmitter. It's actually activating many of these ligand-gated channels. So we have lots of these positive sodium ions entering this dendrite over time. So I'm just going to summarize the sodium ions as this blue positive symbol. And positives repel each other, right? Positive repels positive, negative repels negative. So the idea here is that when this part of the dendrite is filling up with positive ions, those positive ions are going to push each other away, and that actually propels all the positive ions towards the body of the cell, the body of the neuron. By the way, here's the nucleus of the neuron. And so we're still talking about step one essentially here. So step one is still happening. The positive ions are repelling each other. And now they're entering this new space called the axon hillock. So this axon hillock, we have new specialized types of receptors. And these are also a type of sodium channel, but these are called voltage-gated sodium channels. Let's add that to our key. So it looked like an X. So in our key, an X. An X protein channel would look like, it would be a voltage-gated sodium channel. Now, a voltage-gated sodium channel, in contrast to a ligand-gated sodium channel that we saw previously, is going to be activated only when the proper voltage arrives. Now, recall that the inside of the cell is very negatively charged because of what we talked about here. If it's very negatively charged at the resting state, but then we have a big influx of positive ions to this area, that means the positive ions are going to be kind of reducing the negativity of that area of the cell. So essentially, these positive ions are making this part of the cell less negatively charged, or you could think about it as more positively charged. But technically, maybe still it's going to be, I don't know, like around -60, -57 millivolts, something like that. So still technically negative, but it's just less negative. Anyway, once enough of these positive ions come into this area and reduce the negativity enough, then that activates these voltage-gated sodium channels. And I'm actually going to use a different color pen for this so that you can see the difference between the flow of the sodium ions initially and the flow of the sodium ions in our step 2. So let's actually remind ourselves this is going to be step 2 at the axon hillock. This is officially where our action potential is going to pretty much be starting. I mean, we kind of started step 1 up here, but this technically is not considered an action potential yet. The action potential is going to start at the axon hillock. So in this step 2, it's activated. We're going to have sodium ions that are still in high amount outside of the cell. They're going to enter in through these voltage-gated sodium channels and just add to the mix of positive ions. So there's more positive ions than there even were before now. And they're just going to aggregate in very high quantities here. Of course, what does this do to the axon hillock? It's making the axon hillock even more negatively charged than it was before. So what's true of what we said previously is still true here, that the positive ions like to repel each other. So these positive ions that we find in this area are going to be trying to go backwards, but not really very much. They're mostly going to go to the right and travel down here. They're going to start spreading downwards. Now, this is when it gets interesting, I think. So, step 3, I'm going to take my pink pen here because it's going to have to do with potassium ions. Step 3 is going to be regarding this channel. So let's put a number 3 around here. That channel is a voltage-gated potassium channel. So let's add that to our key. Let's see. So it's kind of like a... I didn't do a very good job drawing it there. Oops. Well, voltage-gated, that's my kind of shorthand for it, voltage-gated potassium channel. And so where is potassium highest concentration? Inside or outside the cell? Currently, it's a high concentration inside of the cell. So if this thing were to open up, we should expect the higher concentration of potassium ions that is inside the cell to leave the cell and go outside. And we know that there's a higher concentration of potassium ions inside the cell because of what we talked about up here. So when does it open up? Again, voltage gated, it opens at the right voltage. And the right voltage for a voltage gated potassium channel is going to be something like positive 20 or positive 30 millivolts. I would have to look up the exact number, but something around there, I believe, plus 30 millivolts. So when the inside of the cell actually gets positive enough that it is actually officially positively charged because there's so many positive ions in there, it is going to open this up, and this is going to allow potassium ions that are inside of the cell to leave. And essentially by doing that, the goal of the neuron is to kind of go back to normal, right? Because its resting state was negative, and in order to get back to that negative state, it has to let some positive ions out of the neuron. So the way that this works is that it will utilize the potassium positive ions to let them release from the cell. So step 4 is related to this. So in step 4, we basically have the fact that the neuron is going to go too far in step 3. It's a overcompensation, essentially, by the neuron. What I mean by that is that in step 3, there's actually going to be too much potassium leaking out of the neuron. It does it for too long, and so too many positive ions end up leaving the cell, and this results in the cell being more negative than it was even initially. Now, to imagine that, this would have to be happening a lot of times, right? And so, of course, I didn't draw how many there are in reality, but there would be many, many of these voltage-gated potassium channels, allowing a huge efflux of these potassium ions into the outside of the cell. Now, so it's going to do it too much, and this results in what we call hyperpolarization. Hyperpolarization is polarization past the resting state, or past the resting potential. And you might be asking, well, why doesn't it just close on time? Why doesn't the voltage-gated potassium channel just close up at the appropriate time? Well, this protein is kind of a slow protein. It's specifically, it's slow to close. So it's a little bit of a more complex thing to explain, but I think we'll take care of that in lecture. But for the most part, just think of it as slow to close. So that's why it goes too long. So step 5 is essentially going back to what we talked about initially. And step 5 is pretty much the last step of the action potential. So step 5 is that when those voltage-gated potassium channels finally close up, and things are still really negative inside of the cell, guess what comes to get the cell back to normal? It's going to be our sodium-potassium pump, as well as these other channels, the potassium leaky channels and the sodium leaky channels. They all work together to reestablish the resting potential of the neuron. All right, so we talked about all the basics here of an action potential. I think, in my opinion, the most complicated steps. What I'd like to do now is to think about the big picture here and the rest of it, like what is happening with this action potential, the rest of this area. So action potentials happen in kind of these segments. So we just looked at a full action potential from the axon hillock to this segment of the neuron, specifically to that section of the axon. But there has to be the action potential occurring also in this next segment, and then in the next segment, and then in this next segment. How does that happen? Well, these positive ions that get repelled from the previous step are going to travel down as they get repelled by the positive ions behind them, and they're going to enter this area. And then, as you might be able to guess, by all of that positive ion entering this section, that is going to activate the voltage-gated sodium channels, firstly, adding even more positive ions, right? So we're even increasing the load more, so more and more positive ions. And then once this section of the neuron gets so many positive ions that it goes to like positive 30 millivolts, then that will activate the voltage-gated potassium channel. And then it can begin the process of reestablishing itself at its resting state. And this just keeps happening. So all those positive ions are going to go to this part and then do the whole thing over again. I'm not going to draw this segment, but it's going to be the same as the previous segment. Those positive ions will repel over here. And this is where things are a little different finally. So at the end of the axon, we have our axon terminal. Once again, we have our synaptic vesicles with our neurotransmitters. So we know that there are neurotransmitters inside of that. This thing in the membrane is called a voltage-gated calcium channel. So I'm adding that to the legend. Voltage-gated calcium channel. And so calcium is not an ion that we've been focusing on, but calcium is also an ion that we find in higher concentrations outside of the cell than inside of the cell. And so this calcium is going to flow down its gradient once, of course, the proper voltage activates and opens that calcium channel. And the calcium is important for triggering the exocytosis of these vesicles to the membrane. So without calcium, you don't get these vesicles fusing to the membrane. You don't get neurotransmitters released into the synaptic cleft. So this is pretty much the last stage of the full action potential throughout the neuron, is that you get that calcium influx in order to release the neurotransmitters. So let's go ahead and draw those neurotransmitters being released. Let's assume everything happened, action potential happened successfully, calcium went in, some intermediate steps occurred in this area that causes the vesicles to fuse. And then of course, this is another neuron. Here's a dendrite of that neuron. This would have receptors on it too. This would have those ligand-gated receptors. Sorry, not receptors. These ligand-gated sodium channels. And then the process can start all over again in this next neuron. Okay, so let's put this all together in a graph form. I think the graph is what really tricks most people, so I'm saving it for last on purpose. I put the graph on the bottom of the worksheet here. And so on this graph, we're basically graphing the millivolts or the voltage of the across the cell membrane over time. So for reference, and let me zoom in here. So for reference, our resting potential of our neuron, or what the inside of the neuron is in terms of voltage, is negative 65 millivolts. So what you can do is you can put, I don't know, like a dotted line or something. And that dotted line represents the negative 65 millivolts, which is our resting potential. Then there is also going to be what we call a threshold potential, which I'm going to explain in a little bit. And our threshold potential is going to be just a little bit above that, still in the negative range. So threshold potential. And by the way, time, so if we're thinking about how fast this is happening in a part of your neuron, then the middle is somewhere around 2 milliseconds, or 2 ms. And then by the end of it, it's about 4 milliseconds. So that's kind of our time scale that we're thinking about. Our step 1 was right in the beginning, so our step 1, this is when we had those synaptic vesicles. Which I realized we never really actually technically labeled it on the diagram, so I apologize. Let's just add it there. Step 1 was up here. So step 1 was when that neurotransmitter bound that ligand-gated sodium channel, and we have just the beginning of those sodium ions entering the dendrite. So right at the beginning, there's not really an increase in the, there's not really too much of a change in the voltage of the cell. Not quite yet, anyway. In step 2, however, step 2, we had the activation of voltage-gated sodium channels, and that created a huge influx of more positive ions into the axon hillock. And so that huge influx of positive ions is pretty much represented by this increase from the threshold potential onward. So I'll use my purple pen because we used purple to represent those sodium ions. So a 2, and then everything, so from the threshold potential to the tippy top here, that would be step 2. All that, the cell getting increasingly positive in millivolts, right? Increasing, 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 increasing as those positive ions enter the cell. So what's cool is that that step 2 only happens if you reach what's called threshold potential. That is the voltage that is required to actually activate those voltage-gated sodium channels. What happens if you don't reach threshold potential? So, for example, visually here, what happens if not enough neurotransmitter was released and not enough of these ligand-gated channels opened, and so it was trickling just a little tiny bit of sodium down here, and that sodium migrated and entered the spot? If not enough blue sodium ions from the outside in the first step came in, if there's not enough, then it's not positive enough to activate the voltage-gated sodium channels, then we don't actually get an action potential. Everything would just stop at that point and the cell would reset. So what we call that is a graded potential. So different from an action potential, that's called a graded potential. So let's see, where are we? Okay, so we're now at the tippy top of this graph because the sodium influx has made the inside of the cell very positive. So step 3 starts right at the curvature here when things are going down. That is going to be when the positive potassium ions are leaving the cell. So we can say the beginning of step 3 is from here down. And step 3, we can kind of say it goes to about here. So from like the middle here to about there. So remember, these voltage-gated potassium channels are slow to close, so they're kind of overcompensating to get things back to normal. And so too many positive ions flow out, which results in more negative cell than it used to be at resting state. So this is what that dip represents, is that hyperpolarization. Hyperpolarization. So that is our step 4. So when it's hyperpolarized, and then lastly, things are going to creep up back to normal. It's gonna get more and more positive until it hits -65. This is our step 5, where our sodium potassium pumps are going to work in conjunction with those potassium and sodium leaky channels. Step 5, bringing things back to normal, back to resting potential. So that's all the complex stuff there. In case you're curious about some of these other structures, these guys here are the Schwann cells. The Schwann cells are the cells that coat segments of the axon in the peripheral nervous system, and they pretty much make up the myelin sheath, which is going to insulate the neuron. And by insulating the neuron, it's going to allow these action potentials to go faster down the neuron than if it was uninsulated. In our body, we do have neurons that are uninsulated or unmyelinated, but that's for usually a specific reason. But for our purposes, let's think of neurons as insulated with myelin, which comes from Schwann cells. And then it's only in between the Schwann cells is where that action potential is kind of happening. So these are the in-betweens, and we call them nodes of Ranvier. Nodes of Ranvier. All right, hopefully this was helpful to everybody, and cool, see ya.